Good morning, everyone. Thank you for joining us this morning for the first in our webinar series for the iCell Neuron product. My name is Rachel Lanis. I am a technical applications scientist at Cellular Dynamics International, and I work primarily with the iCell neurons. Today we will be covering the iCell Neuron product in a general overview of the product and functionality of the cells. And the iCell neurons, again, are functional human neurons derived from induced pluripotent stem cells. And before we begin the presentation, which will be recorded and available in future dates on the website, I'd just like to go over the process of asking questions. Questions can be asked at any point throughout the webinar. However, they will all be addressed at the end of the presentation. And depending on how much time is left, we will be able to follow up with any questions that come in that we don't address at the end of the webinar. So as we, if you look at your desktop, you'll see at the top in the center, there's a little tab that you can scroll over. And once you place your cursor near the top of that screen, you will see a display in which there are different icons. And we have a Q&A icon to the right. And this, if you click on this, we'll call it the text entry field. Once you have this open, you can enter your questions into the bottom field and press send. And once again, these questions will be answered at the end of the webinar and by follow-up email. So now, beginning the presentation, I just want to go over an outline quickly. The iCell Neuron presentation will follow this flow. We'll go over a brief introduction of CDI as well as the iPS cell technology. Then I'll go into a few slides on iCell Neurons Characterization, and then we'll conclude with the usage of iCell neurons in various applications, including toxicity testing, functional assays for discovery, and disease modeling. CDI is the world's largest producer of human iPS cells and iPS cell-derived cell types. We really have three core competencies that we utilize in our company. These are the creation and culture of human iPS cell lines. This really is not a trivial fact. We are able to, or we have developed a procedure in which we are able to take a very small blood draw, a very non-invasive process, and are able to generate iPS cell lines from any background, be it a healthy or a diseased source. And the culturing of these lines is something that we have developed a very good process of doing at a high level, which allows us to manufacture at very high levels. We also have a high level of experience in genetic engineering of the iPSC lines, in which we are able to introduce lineage and pathway-specific markers, allowing for the differentiation process to be achieved, as well as any introduction of customer genes of interest into our iPS cells that for use in endpoint assays in your hands. Finally, we're able to manufacture human tissue cells from all three germ layers, and this is something that we're able to do at industrial levels. There are many labs that are able to generate iPS cells and differentiate these into terminal cell types. However, our process is such that we are able to do so at a high level and in a very reproducible fashion. CDI was founded in 2004 by Dr. James Thompson, who was the first person to isolate human embryonic stem cells in 1998 here in Madison, Wisconsin. Dr. Thompson currently serves as the Chief Scientific Officer at CDI and is involved in the regular processes and development of products at CDI. We currently employ approximately 110 people here in Madison, and together that combines over 300 years of experience in the stem cell field. At CDI, we have three key characteristics for adoption. These are quality, purity, and quantity. Our products exhibit the key cellular characteristics and recapitulate the normal human biology. Our iCell neurons will look, act, and function like human neurons. Again, we do this in a reproducible manner where our manufacturing process will yield consistent lot-to-lot, so we eliminate that worry of lot-to-lot variability. And also, our products have known and relevant genotype. The purity of our cells is extremely important as these are terminally differentiated, non-proliferating cell types. In our manufacturing procedure, we have many non-target cell types that are found and are proliferating. We have developed this process to eliminate those non-target cell types over time and maintain a high purity throughout the procedure and in the cryopreserved final product that you will receive. Our product spec for neurons is greater than 90% purity, and in most cases we see often greater than 95, around 97 to 98% purity. And this is something that we are able to do so in our process and in our endpoint cultures through the use of our iCell neurons maintenance medium. And finally, quantity. Our manufacturing procedure has been developed to yield a sufficient number of units to support any high throughput screening process and safety testing. As an example, our current iCell cardiomyocytes manufacturing procedure is capable of yielding greater than 1 billion cells per day, and that is pure cardiomyocytes. The current Cellular Dynamics product portfolio consists of the iCell cardiomyocytes, which were launched in the end of 2009, and endothelial cells and neurons, which were launched late in 2011. Hepatocytes, which are anticipated to launch this year, and we also offer a custom iPS cell service where we can engineer iPS cell lines from any source that is provided and also offer terminal differentiation of those iPS lines as well. In the next few slides, we will focus on the general characterization of the iCell neuron product. And I'd like to begin by just going over the product format. Again, iCell neurons are human iPS cell-derived neurons, and they come cryopreserved and ready to use, fitting into any workflow and are available to store in your liquid nitrogen tank and are ready to use at your leisure. Again, they're terminally differentiated. They will not proliferate. They're not a cell line. What you get in a vial is what you will get on your plate. And they come at very high levels of purity. Again, they are guaranteed to be greater than 90% pure, but oftentimes will be found at 97 to 98% purity when you receive them. They're platform agnostic and compatible with numerous existing assays and instrumentation. They come as a complete product solution that includes the cells, iCell neurons maintenance medium that has been specially formulated to maintain purity over time, as well as the protocols for general cell handling as well as other potential applications. And they are, again, produced in very large quantities to meet the demands of any customer. Some of the general characterizations that we have done of the iCell neurons include examining morphology. As we can see from the image to the right, our cells display both bipolar or multipolar neurite outgrowths. We have stained for several different molecular markers, including the general neuronal markers, beta-3-tubulin, MAP2, as well as some subtype-specific markers, VGAT, VGLUT, and tyrosine hydroxylase. And we have examined the functional characteristics, including electrophysiology, as well as neurite outgrowth. Our iCell neuron population consists primarily of a mix of GABAergic and glutamatergic with a very small amount of dopaminergic neurons. I just want to point out the image to the lower right in this screen. It's 15 conical tube containing a 5 mil pellet of pure neurons, which is approximately 4 billion pure neurons in this pellet. And this really speaks to the capabilities of our manufacturing procedures. This comes from a single production run and just, again, really reflects our ability to produce near unlimited quantities of cells. In this next slide, we really focus on the purity of our cells over time, as well as the development of the neuronal network. So we see in our day 7 post-thaw ICC staining to the right, this is our metric for determining purity. We see nestin negative and beta-3-tubulin positive, and you can see even at 7 days post-thaw, these cells display no nestin staining. Our day 1, day 5, day 19 images show the development of the neuronal network over time, and we see that within 24 hours, the cells have nicely attached to the surface of the plate and have already begun to develop the neuronal network. And this is visible within the first few hours, actually, of planing your cells. However, I recommend highly not looking at them until 24 hours and allowing them to really acclimate to the plate. But this network will, begin to develop over time and will continue to progress and evolve and become more intricate as time progresses. As we can see in day 19, it has become very detailed. And our purity is then reflected in those cultures at these time points. And we see on day 1, we're at 98% purity over time, and at day 19, we've maintained that level above 90% pure at 91.8. So our cells, once again, display a highly pure population of beta-3 tubulin positive, nestin negative, and have display the morphology of mature processes. Now that we've demonstrated that our cells are pure and have the proper morphology, we want to determine what subtypes are found within our population. And from the staining, we can see that we do have a mixture largely of GABAergic and glutamatergic neurons, where we've stained for VGAT and VCLUT, and we see the nice punctate staining of the vesicular transporters. And we also have a staining of GABA to the right to show the mix of this as well. And we have done some staining with tyrosine hydroxylase, as I previously mentioned, and we see just a very, very small amount of dopaminergic neurons, less than 1%. And this population makeup is very typical of the forebrain region. Now, in the next set of slides, we'll cover several different applications and platforms on which our cells can be used. We begin with toxicity testing and then move into several other aspects. In this slide, we focus on the use of iCell neurons in cell-based assays. And we can see that in the top graph, the treatment with known toxicants staurosporin and chlorpromazine yield a dose-dependent decrease in drop in viability at the expected physiologically relevant doses. And then in the graph below, we see that treatment with the excitotoxicanic acid yields the expected results and nice dose-dependent response as well across multiple lots. And this is non-normalized data, but it really speaks to the consistency of our products in that multiple lots will yield the same approximate EC50. So again, our cells can be used in cell-based assays to show reproducible and cell-specific results across multiple lots. Now we can move to study our cells in mitochondrial membrane potential on high-content imaging. The work on this slide was done in collaboration with molecular devices. Mitochondrial membrane potential depolarization has been known to be an early predictor of excitotoxicity as well as oxidative stress. And what we've shown here is when staining with the mitochondrial dye JC10, we can see that in our control culture, we have the nice orange staining throughout the population. Whereas in contrast, with treatment of 3 micromolars of antimycin A, we no longer see that nice staining throughout the culture. In the graph to the right, we then show a nice dose response with the treatment of antimycin A as well as the valinomycin. Again, this was done on the molecular devices ImageXpress microreader along with their MetaXpress software. The next 2 slides will focus on some of the electrophysiology functionality of our cells. On this particular slide, we address the ion channels present in our cells, and we have both the sodium, potassium, and calcium channels. And we see the control activity of those channels on the far left of this panel. And then we've treated each channel with known antagonists. For sodium, we've treated with TTX, potassium, TEA, and calcium, the L-type channel blocker, nifedipine. And we can see that we have, on the far right, in dose response shutdown and blockage of these ion channels. Now, in this slide, we address the postsynaptic receptor functionality of our cells. And in the top panel, what we've done is added a prolonged treatment of the excitatory agonist canic acid, and we see a nice depolarization into the cells, while then a treatment with the antagonist CNQX and AP5 for the AMPA kinase and NMDA receptors blocks that activity. Similarly, treatment with glutamate, another excitatory agonist, causes not quite so dramatic an increase of depolarization. However, we do see a little bit of a depolarization and, again, blockage with the antagonist CNQX and AP5. Alternatively, when we look at the inhibitory currents, we see that treatment with the inhibitory agonist GABA causes a hyperpolarization, and then treatment with the antagonist bicocylene blocks that activity as well. So this data really demonstrates that we have the 3 different receptor types present in our cells and that they are all functional. So the eFuse data here shows that our cells can be used for studying both ion channel and synaptic responses. On this next slide, we will take a look at the neuronal networks and studying neurite outgrowth of our cells. In these particular graphs, what we're seeing is that treatment with known toxicants, antimycin A, mitomycin C, and staurosporin at increasing dosages cause a disintegration of the neuronal network. In the graph on the left, this is measured by the mean neurite outgrowth. So this is the length of the neurites relative to each cell. And the graph on the right, we look at the total number of branches per cell, and we see a rapid decrease at the expected doses. Now, these images show a visual representation of the treatment with mitomycin C here. We see the control with a nice neuronal network that was previously shown to develop very early on, and increasing doses really disintegrate and eliminate that neuronal network. These cells were allowed to be maintained in culture for approximately 48 hours prior to a 72-hour treatment with the compounds. Finally, the high-content imager in collaboration with Molecular Devices was used to study live cell toxicity. And here we see that we've treated with 5 or so different compounds, and once again, see the expected decrease in neurite outgrowth at the relevant doses. And we're staining with calcium AM and can see that with increasing doses of methylmercury, we are losing that viability. We have also looked into transiently transfecting and transducing our iCell neurons, which would be of great interest for anyone looking to introduce reporter genes or other genes of interest into our cells. And we can see from the graph, the image at the top, that we are able to transfect our cells with GFP. And this will follow a very standard workflow of thawing and plating our cells, transfecting with the reporter gene, and inducing and measuring the activity. Again, then we've also transduced our cells with the AAV and RFP, and we can see that over a period of roughly 3 weeks in culture, we increased our efficiency up to 75% with an MOI of 60,000. And again, we see the images on the right show that increase in transduction, where at day 25 and 60,000 we see a nice red fluorescence. We've also determined the levels of the tau protein and A beta proteins in our cells through collaboration done with mesoscale discovery. And what we essentially are able to see is that our cells do produce normal levels of both tau and beta amyloid proteins that are typical of a normal adult. And what we've done in these experiments, as with tau as a lysate assay, we've compared multiple lots on multiple days. Also with the beta amyloid protein, this is a conditioned medium assay. We've compared multiple lots as well as multiple densities across multiple days. And what we are seeing here is that the level of expression is stable over time from 7 days in culture to 14 days in culture, as well as reproducible between multiple lots. And with the beta amyloid, again, the doubling of that density really doubles that level of expression. And since we are expressing the expected normal levels for an adult, these cells can then be used to study diseases such as Alzheimer's as well as other tauopathies. This next slide is really just a nice way to capture different papers and publications that have utilized the IPS technology to generate IPS lines from diseased patients and terminally differentiate those into cell types which then reflect the expected disease phenotype. And we can see spinal muscular dystrophy, the cell phenotype reflected is selective motor neuronal death, schizophrenia. We see decreased synaptic density. Alzheimer's, there are increased levels of the amyloid and tau proteins. And this then speaks to the capability of our cells to be utilized in recapitulating disease phenotypes in the dish. Now, to summarize, we've shown that stem cell technologies provide an unprecedented access to human biology. Again, we are able to generate iPS cell lines and terminal cell types from really any tissue source. We've developed a novel commercial platform for reprogramming these lines and differentiating them into neurons. Our iCell neuron product is a mixed population of GABAergic and glutamatergic neuronal subtypes. Again, this is very typical of the forebrain region. They're terminally differentiated human iPS cells and exhibit the expected neuronal characteristics and functions of humans. And the iCell neurons provide a standardized model across multiple testing platforms and assay, and they are reflective of relevant human biology. And with that, we'd like to thank you for joining our webinar, and we'd like to start taking any questions that you might have. So our first question is asking, what equipment was used to create the EFIS data? This was all done on using single-cell patch clamp. The exact vendors for the equipment I don't know or have in front of me, but that is certainly information that can be provided. So I can follow up with you on that. All right, and another question that we have is, can these cells be cultured on matrigel? That's actually a great question. We haven't discussed the extracellular matrix that we typically use with the neurons, but it is the recommended matrices is a PLO laminin double coat. And this is something that is gone over, and there's a protocol for it in our user's guide. We have tried matrigel with our cells. They really do not adhere very well to this. They will sit down within a day, within the first 24 hours. However, media change on day 1 will blow off all cells that are attached. So it is a matrix that we do not recommend using. All right, another question we have is, can we use antibiotics in the media? Now, we don't typically, well, our media is not provided with antibiotics in the medium, but it is certainly something that can be added. And in fact, all of our EFIS data is collected using antibiotics in the medium because it does require exposing the cells to open air conditions. So certainly antibiotics can be added to our medium without impacting functionality. All right, another question we have is, how long can you maintain these cells in culture? Most of our functionality data and characterization has been collected between 7 to 21 days, so 1 to 3 weeks in culture. However, we have taken our cultures out for quite a bit longer than that and maintain them for 3 months, 4 months with regular feeding schedules of every 3 to 5 days. And we don't have functionality data at these later time points. However, morphologically, the cells do maintain the neuronal network that we've shown. All right, we have a question also from someone who came into the webinar a little bit later about the purity of the neurons. So just to reiterate, the purity of our neurons is always going to be greater than 90%, and that will be when you receive them. In most cases, much greater than that, above 95%. We often see around 98% purity, and that is what you would expect to receive in your shipment. Is a population of around 97-98% pure neurons, and that spec will be listed on the certificate of analysis as well. And we have another question asking, can these cells be put back into suspension after several days of plating for automated patch clamp? This is something that we are not working on internally, but we do have collaborators working on this process. And as that data comes back and protocols are developed, we would certainly be able to provide information. But at this time, we have not done that internally. Another question coming in is, how do you get rid of the astrocytes percentage Well, our manufacturing process, again, is developed to purify our product to be a neuronal population. So we do not have any astrocytes present in our final product, and this is done through the proprietary methods of our purification process from IPS to the final product. All right, final question that we have is, how long can these neurons survive in culture in our hands? And, you know, again, this, I just spoke to this a moment ago, is that we do all of our functionality testing typically within 3 weeks of culture. However, we have taken them out much longer and maintained a nice viable cultures for several months. We do have some customers who have looked at longer-term assays as well, taking them out 5-6 weeks in culture. So the cells really, with a regular feeding schedule of every 3 to 5 days, can be maintained indefinitely, and so that you shouldn't have any problems taking them out for longer term. All right, with that, thank you very much for joining, and if you have any further follow-up questions, please contact CDI's application support, and we'll be happy to follow up with you.